Well, thank you very much. I think we should make a start. So could I start by welcoming you all to this special distinguished lecture symposium on modeling in surface science and catalysis. Very good to see you all here, and also welcome to those who are joining us online. Modeling in surface science and catalysis is now very, very extensive. Modeling tools are absolutely indispensable in these fields. And in fact, I think if you look at the majority of contemporary papers published in catalysis and surface science, including those that are predominantly experimental, you will find that they have quite a strong, significant modeling component. So it's a very active and important field. But it's a field where a great deal is happening, a lot of developments, a lot of technique developments, and lots of exciting new applications, but also with improvements in technique, the possibility of revisiting some old and long-standing problems. Well, we're going to hear from a number of speakers this afternoon. I'm very grateful to them all for joining us today and sharing their expertise. But a special thanks to Professor Joachim Sauer, who has worked in this field for many years and is a leading international expert. And Joachim is also a very good friend, both of UCL and of the Thomas Young Centre and of the UK, both catalysis and modelling communities. So as I say, it's a real pleasure to welcome you all here this afternoon. Good to see such a good turnout in person. And I'm now going to pass over to Jochen, who is the current chair of the Thomas Young Centre, who is going to introduce the speakers. So over to you.
Okay, thank you very much, Richard. It's a pleasure to see you all here after almost 2 years of doing the seminars online. It's great to see you here for the first time in person. So it's the first post-pandemic seminar. And so lots of things has happened in the Thomas Young Centre, especially we've got our website got a facelift, so please check it out. And for those who join us for the first time, please feel free to sign up to our interest groups. And also please, get involved if you'd like to get involved in running the center. So you're very welcome, and a lot of information you find on the website. So it's a really great pleasure here to sort of chair this symposium today. And our first speaker is Dr. Tom Keal here from the Dasbury Laboratory. Tom is a principal scientist here in computational chemistry. He did a PhD some time ago in 2005 with David Tozer in Durham, trying to develop the exact exchange correlation functional. And he then moved to Germany to work in Walter Thiel's group in the Max Planck Institute. And he also worked there on excited states in biomolecules. After that, he joined Sherwood's group in Dasbury in 2008. And I think since then you have been busy developing the ChemShell code and especially taking care of the QMM functionalities in here. And this, we're going to hear about the latest progress today. So please, Tom, the floor is yours.
Thanks very much, Jochen, and thanks very much to the organisers for inviting me to speak today. So yeah, I'm going to talk a bit about some of the recent development and application work that we've been doing in the ChemShell software that we developed at Dasbury for QMM calculations. So if we start the video. Okay, I stand very still. Let's get rid of that as well. So, is that working? Yeah, I'm just trying to minimize the bits and bobs. It's gone on my screen. Oh, it's this one, isn't it? That's why. Yeah, it's okay. So many of you might be familiar with the ChemShell code. If you're not, I've got a couple of slides to recap what it's all about. So ChemShell is a computational chemistry environment, so it's controlled by a scripting interface, and it's general purpose package that's suitable for any kind of chemistry calculation. But its particular focus is on combining quantum and classical calculations, so multiscale QMM simulations. ChemShell, you would say, is very well established code. It's now over 25 years old. And over that time, it's been used principally for biomolecular modeling, so DNA, proteins, particularly enzyme reactivity, and also for materials chemistry, for things like heterogeneous catalysis, so reactions on surfaces and looking at defects in bulk systems, for example. And key to this philosophy of ChemShell is flexibility. So that's what allows it to be used in a range of contexts. From users' perspective, it's a matter of writing Python scripts, which will control the ChemShell code. And then ChemShell underneath has a series of modules which do different tasks related to these calculations. So it'll take care of all the data bookkeeping, for example, the data structures with geometries and results of the calculations. It'll do geometry optimization as a module for that. It does electrodynamics. It has sort of guided workflows and more sort of helping to do more sophisticated calculations. And it's got quite a sophisticated parallelization framework for HPC platforms. The key to everything is the module that does the QMM coupling calculations. And what you need to know is that ChemShell doesn't do any energy or gradient evaluations itself. It calls out to external codes to do that. So it acts as a wrapper around external quantum and classical codes. So it's a range of interfaces to the code, for example, in the box on the left for quantum calculations. And then on the MM side, we typically use either DL poly or GULP, depending on the system, and also a bit of NAMD for doing biomolecular calculations, which I'll talk about later. So that's a very brief introduction to ChemShell itself. And now I'd like to talk about a couple of case studies that we've done recently, one in the biomolecular side and one on the material side. And the first example I'm going to talk about is a biomolecular case study. This is work that Catalysis in my team did together with Jin Yong in our department and our experimental collaborators at the University of Essex. And what we wanted to look at was the potential for combining QMM calculations with a serial crystallography method called MSOX. And MSOX stands for multiple structures from one crystal. And as the name suggests, what we do is we fire X-rays at our protein crystal, and we record multiple structures over time. And the reason this is useful is because the X-ray, when it interacts with the protein crystal, it interacts with water in the crystal to generate solvated electrons, and they are then available to initiate redox reactions that take place in your enzymes, so it can start a catalytic cycle. And so when this interaction occurs and the redox reaction takes place, the structure that you see when we do the crystallography will actually change, and then we can create what we think of as a catalytic movie, where we can see the reaction proceed in the crystal itself. And this is really interesting from a QMM perspective, because rather than just taking a single static crystal structure, which you typically do for a biomolecular QMM calculation, we actually have a series of structures, and these represent fixed endpoints of the reaction that takes place in the crystal. And they can constrain the possible reaction paths that can take place in the model. So these we can use as both endpoints and intermediates in our QMM calculations, and it simplifies the task of identifying the reaction mechanism. QMM in turn can then provide additional information that experiment can't. So particularly on things like preferred oxidation states for a metal center, protonation states for the surrounding amino acid residues, and of course more details on the reaction mechanism as it actually takes place. And the example case study that I'm going to look at is an enzyme called the Nitrite Reductase. This enzyme is found in bacteria, and it plays an important role in the global nitrogen cycle. You can see on the top left sort of schematic of what happens in the nitrogen cycle. And the red arrow there is the reaction that we're interested in, which is converting nitrite and reducing it to nitric oxide air. And an example of an enzyme that does this is the one we're looking at, which is ACNR. So this is a trimeric protein, and each monomer contains 2 copper centers. One, the type 1, is involved in the electron transfer process that initiates the reaction. And the second center, the type 2 copper, is where the catalysis actually takes place. And this is a 1 electron reduction involving 2 protons coming in, and it forms NO plus water. So what we want to know basically is what is the mechanism for this reduction. And by combining the QMMM with the MSOCs, we first start with MSOCs reference data. So this is experimental structures that have been taken over time. They were collected at 190 Kelvin, but we see similar things at different temperatures. And what we see experimentally is, first of all, a nitrite bound to the type 2 copper center in a so-called top hat orientation. So that's with the nitrogen pointing upwards. Then later on, during the course of these structure collections, we see the orientation change to a side-on orientation. This is where all 3 atoms of the nitrite are at similar distances. And then finally, later on again, so after another series of snapshots, we see the NO product. And so then it's the task of QMMM to figure out what is going on between these different snapshots and what do these different orientations and products represent. So in terms of how we set up the QMMM calculation, it's slightly different from how we normally do a biomolecular QMMM calculation. The basic underlying principles are the same, so we use electrostatic embedding. We do a sort of typical QM region of the metal center plus the important surrounding amino acids and any water molecules in the immediate. We use Orca for our QM calculations, which is very good for biomolecules, and standard functionals and basis sets. What we don't do when we're comparing with the reaction in the crystal is solvate the system. So what we normally do is we solvate it with classical, with a sort of classically optimized water ball, and then equilibrate it with classical MD, and then take snapshots from that. We don't do any of that. We just take the initial protein crystal structure, clean it up, add the hydrogens and so on, but use that as our starting point for these calculations. We can then later compare with a sort of conventional solvated system, and that's what we do. So we compare these 2 ways of modeling, but it's not our primary aim. So our primary aim is to model the reaction as it takes place in the crystal. So what did we find? First of all, we looked at the first 2 images. So we looked at the top hat NO2 and the side-on NO2, and tried to figure out what is it that's driving its orientation change. And what we found is that this is all to do with the copper oxidation state. So if we're in copper 2, we find that we can get a top hat minimum, but if we're in copper 1, we can't. And similarly with the side-on NO2, if we're in copper 2, we can't get that, but if we're in copper 1, we can. So it's pretty clear from the calculations this change of orientation is related to the reduction from copper 2 to copper 1. What we in fact find when we try to do the top hat orientation with copper 1 is that it forms a barrierless path to another orientation, which we call top hat monodentates. And this is a bit like top hat, except it's sort of skewed. So one of the oxygens is much closer to the copper center than the other. And then we can use that as a starting point for nudge elastic band optimizations to find the path to the side-on orientation, and we see that there's actually just a small barrier to that side-on NO2 minus forming, and that's probably why we don't see this transient state experimentally. So this is all clear, and we see similar results then when we run it in the solvated QMMM setup as well. The second thing we need to figure out is what's going on between the side-on NO2 minus to the NO products. And here it's a bit more complicated because this is when the protonation states take place, steps take place, and we had to consider a lot of different protonation states of surrounding amino acid residues when we were looking into this. And some combinations didn't work, some did, and it all got quite messy. But in the end, we find a reasonably simple process that takes place. First of all, when the NO2 minus is first protonated, there's a barrierless scission that takes place, resulting in an OH minus and the NO down to copper. And then a further proton comes in from hist. So we've assumed that both the histidine and the aspartate are protonated, and they form a kind of hydrogen bonding network, which allows more protons to come in. And then following this, we get water formation. bound to the copper and NO, and then the water leaves and results in the final copper NO solutions. This all makes sense. And then on the right, you can see the full reaction scheme, starting from the top at NO2 minus bound to copper 2, and then ending up with our copper NO at the end. So this all makes sense. There is one complication, which I haven't mentioned so far, and that is to do with the second snapshot. So where the side-on NO2 minus is observed in experiment. And what we find actually with our DFT QMM calculations is that the energy surface isn't that simple when we simulate it. There's actually a whole range of different orientations of nitrite that you can see, not just side-on, but also a reverse side-on, where it's pointing in the opposite direction, and an end-bound, where the nitrogen is pointing towards the pole. And this causes no end of grief when we're searching for the reaction mechanism, because especially for the solvated system, depending on what snapshot you take, you actually get different ordering of these states. What we can say is when we're looking at the reaction in the crystal, these other states are not observed in MSOX at all at any temperature. They've never been observed. And that's what we mean by this MSOX guided QMM. We can say for sure that these don't exist, and therefore that constrains the reaction path that we can take in the crystal. The problem, of course, still is that we can't make that same statement in the solvated system because we're not then got the experimental comparison. And so we'd still like to be able to understand what it is that's causing the discrepancy between the simulation and experiments. And we did a whole host of fairly straightforward things like messing around with the DFT setup, the basis sets, doing simple entropy corrections of vibration frequencies, or all this kind of thing. None of those things helped to the extent where the ordering is always consistent. And so we believe that more advanced QMM methods are required, probably some sort of more sophisticated free energy calculations are needed to resolve that. So that's something that we leave for future work. So the second case study that I'd like to talk about is, again, nitrogen chemistry, but this time in a different context of zeolites. This is work that's being done by Jamal, who's a PhD student at UCL, working with Alexei and Richard and in collaboration with us on our current ChemShell development grant. And this is looking at catalysis of zeolites and a process called selective catalytic reduction, which is a very important way of reducing atmospheric pollution from burning fossil fuels. And it gets rid of nasty pollutants like NO and NO2 and converts them into nitrogen gas. And the reason why zeolites are interesting for this is there is a lot of evidence that that they can be a very efficient catalyst for this process. So there's a zeolite called copper SSN13, which is a Chebysite topology, and this has found out excellent activity for this process. But it's quite a complicated reaction. There's various different pathways as shown on the left that it can take, and we need to understand the full catalytic cycle, which involves both the oxidation from copper to Cu2 plus releasing nitrogen, and also the reverse reduction process, which again involves the release of nitrogen with different inputs. So there's quite a lot to understand about it. Again, so Jamal was looking at QM calculations to investigate this, and the way they're set up is fairly similar to the protein case. The key difference is that the zeolites are, because they're extended semi-covalent systems, you need to cut them at some point, and then it becomes more important to reproduce electrostatic interactions that you lose by looking at a non-periodic system rather than using periodic boundary conditions. And to do that, we fit a layer of point charges around the outside to reproduce some of these missing electrostatics. And that's all involved in the setup of the cluster. So the original cluster setup is just a siliceous one. And then once that's optimized at the classical level, you can then introduce the catalytic center, so the aluminium and copper species, and do DFT calculations to figure out where the copper is most likely to go. In this case, I think it's in the 8-membered ring of the active site region. But otherwise, it's a similar sort of calculation. In this case, Jamal uses Games UK, which is particularly good for scaling to larger systems, and a couple of different functionals, and then the Hill-Zauer force field for the MM region. So what we wanted to look at was what would be the effect of solvation on these sites. So because water is produced during the course of the reaction, it's going to be present in the system, and of course ammonia as well is a key player in the reaction. So these could well affect the course of the reaction and how easy it is to make the species along the route. What was found was that in fact, as you add in extra water molecules to the copper center of the active site, it actually increases the distance from the aluminium center, and so this is a plausible means of which these copper sites become mobile, and therefore promote the reaction. And a similar thing happens to some extent, to a lesser extent, with ammonia as well. And we can go a bit further than that and look at the different energies of the different species in the catalytic cycle. And there's quite a lot of different species in there that we need to take a look at. And what we found is two different findings that sort of go against each other. So in the initial steps where Cu plus goes to Cu2 plus, the addition of solvents, so water and ammonia, actually promotes the reaction. So if you look, for example, at the formation of nitrate, you see that's lower in energy when we have an ammonia or water bound. But on the contrary, when you're going back from Cu2 plus to Cu plus, we see the opposite. So when there's an ammonia or a water bound, it's actually harder to make some of those species, particularly the HONO and the nitrosamine after that. So there's a lot of debate about whether the presence of water, for example, facilitates or inhibits the reaction, and you can see clearly there's competing factors there which make it complicated. How long have I got?
2 minutes.
Okay, so I'll go through this quickly. What I'd like to finish with is some of the very recent sort of developments that I've been looking at as part of the PyChemShell redevelopment program. So if you know ChemShell at all, it was originally a program based on the TCL scripting language. Then a few years ago, we did a major rewrite of that to make it Python based. And there was various motivations for doing that. One was that we could, by rewriting the code base, it made it a more modern platform that was suitable for future developments. Python we thought was easier for new users to pick up because it's by this stage it's become much more popular than other scripting languages. It's kind of become the standard of scripting for scientific applications. And also it meant by rewriting it, we could start with a new license that was open source, and that would appeal hopefully to users and developers more than the original. I mentioned very quickly a couple of new features that are available in the latest release. So one of these is QM with periodic boundary conditions. So the case studies I showed before were all using kind of molecular QM codes and based on sort of finite non-periodic models. We can also do periodic QMMM with PyChemShell, and this is particularly targeting metals. So where we really need delocalized description of the quantum region. And so we target different ranges of codes as well. So in the example, we use CP2K as our QM code. Again, we use Gold because that's simple for periodic as well as non-periodic. And the aim here is then to study excited states as well with our probabilities. There's also a new biomolecular workflow. So again, this is looking to go beyond the TCL version of ChemShell. In that version, we just assumed that basically the structure was ready to be calculated with the QMM, and the user was left to provide those sort of QMM ready structures themselves. In the current PyChemShell, we go a lot further than that. So we try and help the user starting from downloading a structure from the protein database, helping with all the preparation by interfacing to the various tools that it can be used for cleaning up and preparing structures, guiding the user through the original, the initial sort of solvation and equilibration stages using NAMD, and then reaching the point sort of together that it's ready for QMMM, which is when we then use ChemShell itself to do the coupling with charm and amber force fields. Finally, very quickly, Jing Chang here at UCL has been working on spectroscopic signatures. And he's implemented a method for the vibrational self-consistent field, which is to do anharmonic IR and Raman. And this is an example of ammonia, again, in a zeolite environment, looking at how the environmental effects change the behavior of ammonia, and looking at the anharmonic effects compared to the harmonic ones. Better wrap up. So just one slide on future work. So the work I've been talking about is all existing projects which are sort of coming to a close now, and there's a few new projects that I'm going to be looking at. One of these is a BBRCC one on enzyme engineering. So this is kind of following on from the MSOX guided QMMM that I was talking about first. This is looking at heme-containing enzymes this time, but again involved in nitrification, and looking to see whether we can change the structures of the enzymes to affect the products there. Another big target is exascale computing. So I'm part of the PAX HPC Excalibur project led by Scott Woodley here. And this is a very large project aimed at preparing all software that's related to particle-based modeling. So it covers everything from atoms to galaxies. But there is a role in it there for ChemShell, and that is looking at complex multiscale workflows, materials and molecular modeling. And the key thing there is to link it up with other MMM codes. So there's a number of code developers involved in the project, and the key task is how these codes can talk to each other. Another project involved with is in quantum computing. So this is a commercial project with Artery Center, which is our sister department that's kind of industry facing. And we're working with them and the National Quantum Computing Center and a number of commercial partners led by CQC to look at how we can integrate HPC and quantum computing together to do embedded calculations. And finally, we're still looking at Catalysis, and we've got a new ECRC project together with Richard and Adrian and a number of experimental groups, the UK Catalysis Hub, to look at the emerging field of hybrid catalysis. And all of these projects couldn't be possible at all without the support of COSEC, primarily the Materials Chemistry Consortium, but also CCP5, CCP Biosyn, which provides a kind of long-term means for support of the code. And really, that's absolutely crucial for development of software like this. So hopefully it will continue for a long time to come. So that's this is acknowledgements of the projects that I've talked about and our funders. And thank you very much for your attention.
Thank you very much, Tom. The session is now open for questions. Please.
This was very nice, thank you. So your multiple structure thing, how does this work? So normally I would take a crystal structure and then I would define my quantum part and I would optimize. And then I would look for a transition structure and I would find an intermediate and I would all optimize. So what does it mean? So you have more crystal structures you include?
Yeah, so we have so we have the three crystal structures that where was it? So these are three different experimental crystal structures, and we use those as fixed endpoints in the reaction path.
So you freeze the environment and optimize only the quantum part.
No, we do relax part of the MM region as well. It's true that we have a shared environment between them. Otherwise, we run into problems with calculating paths between them.
So you have kind of boundaries for all the structures, and in between you optimize.
Yes, that's right. So if we don't have a consistent environment, then that'll create artifacts between them. So that's true. That's a complication of the method. But what we see experimentally is actually there's very little change in the environment between the different crystal structures because all the chemistry is happening in quite a localized site, and so it's not a very crude approximation to then use a consistent environment across it.
And does it need some specific tools in your ChemShell program?
No, we use the standard geometry optimization technique. So we have the neutral acid band method in our geometry optimizer module. That's the kind of workhorse for these calculations.
Richard, please.
Just a quick one, Tom. The experimental data here from X-step studies.
This no, this particular one is not, but yeah, Excel is becoming more and more important. So we can do a single.
Increasing use of Excel with protein crystallography.
Absolutely, yeah. So
a big demand for this kind of model.
yeah, so we can do pretty much the same thing with Excel.
Yes, please.
I have a question about the NP calculation you did on the when there is a change in the position of the antibiotic medium. You say that it is associated with a change in the oxidation state of the center. But how do you determine the oxidation state of the center?
So this one, you mean?
Yes.
So what we did was we took the first snapshot, for example, so the top hat orientation of the nitrite, and we just did an optimization with copper-2, and then we did another separate optimization starting from the same structure with copper-1. And what we found is that if you optimize with copper-2, then you get a minimum that's pretty much the same as the experimental structure, more or less. If you optimize with copper-1, that's when you get this top hat monodentate. So what we envisage happens is that the electron comes in, it reduces copper-2 to copper-1, then it goes from the top hat structure to the top hat monodentate. That's a transient structure because the barrier is so low to go to the sidon, so pretty much straight away it goes to the sidon. And that's how we know that we've got the right couple of observations.
Any other questions? Yes, please.
I didn't get it. How you treat metals? As a quantum system or as some potential?
Yeah, sorry, I rushed through this slightly because I was running out of time. But yes, so the approach we take is one that was developed by Jürgen Mayer and Carsten Reuter a few years ago now. It's quite nice. It's very simple conceptually, but it should work pretty well for the metal system. So what we do is we calculate. So it's applicable to surface absorbate systems. So we have we do a QM calculation, which is periodic, with an absorbate on the surface, and then we do another QM calculation without the absorbate, and we subtract one from the other.
But this means you have to treat the whole periodic system with QM.
No, we only do it in the vicinity of the reactive site. I mean, it has to be large enough to avoid kind of absorbate.
Is there anything else than QM?
Yes, so there's an MM bath basically. So the MM, the gold MM calculation is surrounding it, and that's much larger.
And how does it look like? I mean, embedded atom potentials, or what is it?
That's a good question. I have to check the. It depends on the system, obviously, but yes, I think so. Yeah, I need to look that up.
I have a question. So your QMM implementation, does it support electronically fluorizable protein force fields?
Protein force fields.
Yeah.
We have that in TCL ChemShell. So that's one of the features that we haven't ported across yet. So yeah, there was a development that was done in Walter Thiel's group to introduce Drude oscillators into the charm force field. That is, I mean, in terms of the actual physics, it's almost exactly the same as the shell model potential that we use for materials chemistry. So we ought to be able to have a kind of unified approach to doing that. But it wouldn't, I would say it wouldn't be straightforward in Python ChemShell just yet. So yeah, that's one of the things on our to-do list.
Great. Are there any other questions? If not, then we would like to thank Tom again for great work. Adrian sets his presentation up, I introduce our next speaker. So it's Adrian Mulholland from the University of Bristol. Now, Adrian actually started off as a working as a wine merchant. Is this right? Is this a biography? So maybe you do the wine selection today. But then he sort of went back to university and did his PhD at Oxford with Graham Richards. And that followed then a postdoc with Martin Karplus. So actually we have sort of two alumni from the Nobel Prize winners of 2013. So that's quite nice. And then he started his independent career back in the UK. And he published more than 200 papers, and he was also recently awarded the John Murray Thomas Medal 2020 for his outstanding and innovative work in the catalytic sciences. So I know Adrian already now for more than 10, 15 years or something like this, and he has always impressed me with his sort of work, high quality work, let's say, on enzymatic mechanisms, always bring in always high quality electronic structure methods for the QM calculations. And so he's also working on enzyme design and also on understanding reaction mechanisms and mechanisms of antimicrobial resistance. So I'm very much looking forward to your presentation today, which will be on multiscale modeling of biocatalysts and enzyme design, evolution, and engineering. Adrian,
thank you very much. Thank you, Jochen. Thank you very much to the organisers. It's a pleasure to be here. I guess we're having several, as we've already seen, biological applications, and we'll see more from Edna later. I think the nice aspect of this symposium is the common methodological threads that link different areas of catalysis. This is something that Joachim and Richard have talked about and is exemplified, for example, by the widespread adoption now of QMMM methods in all areas of condensed phase catalysis. That's a good start. So QMMM methods. So QMMM methods became officially respectable in 2013 when we had the Nobel Prize awarded to these guys who've already been mentioned. And of course, why is QMMM useful, or modeling, quantum mechanical based modeling, useful for catalysis? Well, one obvious area is in studying unstable species, transition states, reaction intermediates, and so on, how they're formed and stabilized within systems such as enzymes. So this is a transition state. This is from work of the late, great, and much missed Walter Keal, showing a, do I have a mouse? An OH group in flight here between these two groups, and it's stabilized by a hydrogen bond. And this hydrogen bond only exists in the transition state. So this is a catalytic interaction, which was identified by the QMMM model. And these enzymes, where we know the mechanism very well, provide guinea pigs, tests for QMMM and other methods. I won't go into the long names of the enzymes, but we can use these to test different levels of theory, as Joachim's done in non-biological systems. And what you find from that, not very surprisingly, is that you have to go to quite high levels of QM theory to be able to predict barriers with chemical accuracy. But once you go to high levels of theory, so where appropriate, for example, a local coupled cluster method, if I can say it, you can indeed predict barriers that are, roughly speaking, within chemical accuracy of experiment. So you can predict a barrier to a reaction in a system such as an enzyme with as much accuracy as one can measure the experimental kinetics. And that means that these calculations can indeed be predictive of mechanisms. We can rule in and rule out mechanisms, as Tom was just talking about. It turns out that those particular calculations used the local approximation zone, which are quite difficult to apply. A more straightforward way to approach chemical accuracy, we find, is to go to another level of multiscale in which we embed ab initio treatment using projector-based embedding within a density functional region, within a wider molecular mechanics region. And why is this useful? Well, it allows for a straightforward and rigorous application of methods such as coupled cluster in a practical context. Single point calculations, for example, on structures from a DFT reaction pathway. And that's important because, as is well known, if you do calculations with different functionals, you'll often get rather different answers. So this is going back to one of those guinea pig enzymes. This is charismate mutase, so a Claisen rearrangement reaction. And what you find here is that the barriers are really significantly different with different density functionals, which is not wildly surprising, but it means that those calculations are not in themselves predictive and could not be used to predict the mechanism with certainty. However, if we use projector-based embedding, embed in this case, an SCS MP2 treatment within the DFT region, within our QMMM calculation, we can remove the density functional dependence entirely. This is the SCS MP2 in DFT QMMM calculation. And you can see that all the methods, therefore, then agree. So this is there are alternative approaches to going to higher levels, such as those developed by Frank Naser. This is important, this high-level treatment is important if you want truly predictive calculation of variance. On the other hand, we can't always do coupled cluster calculations, and not only when you have chemical complexity or lots of metal ions and so on, but in cases where we want to make rapid predictions of activity, of catalytic activity. One such application is in the area of antibiotic resistance. This is the so-called silent pandemic, the widespread dissemination of resistance amongst bacteria to the antibiotics that are used every day in the clinic. And that includes resistance to what were last resort antibiotics, such as carbapenems. And if you're unlucky enough to get a gram-negative bacterial infection that is carbapenem resistant, there may be no effective treatment for that bacterial infection. And that is not only threatening to surgery, it also threatens much of modern medicine in transplant treatments, cancer therapy, and so on. So what has happened over the years is that enzymes have arisen, so that antibiotic resistance has been around for longer than clinical use of antibiotics. But what has happened over time is that enzymes, the old enzymes, these up here, I'll go into all the names, which could break down penicillin, for example, but were inhibited by carbapenems, have been replaced by more recent variants, which can break down carbapenems. So that and the difference between them is that they deacylate faster. So they break down the covalent complex formed during catalysis much more quickly. So here we want to be able to discriminate between those enzymes that can break down carbapenems and those that cannot. And we're going to the opposite end of the QMMM spectrum here and using low level of theory. So we're using an SCC DFTP method within QMMM molecular dynamics simulations. And we can indeed distinguish between the enzymes that have a low barrier to deacylation, so the carbapenemases, and those that have a high barrier to reaction. And if we do a full QMMM free energy surface calculation using umbrella sampling, it takes us about 20 days per enzyme to make such a prediction. And the barrier, you can see we try to estimate the statistical uncertainty from repeated simulations. So that's good, but it's not fast enough really to be competitive with experiment. So how quickly can we do such a calculation? Well, if we reduce the computational demands by either just focusing on the minimum energy pathway, we can get down to about a couple of days. If we go down even further, so reducing the sampling, we can now make predictions of QMMM free energy barriers for an individual enzyme in less than 4 hours. So that means that you can do the calculation, roughly speaking, as quickly as you could do the experiment. And of course, the calculation is rather cheaper and can be set up rather more quickly. And that means that these calculations, QMMM calculations, can be used a priori to predict the activity of enzymes against different antibiotics, which may be useful, for example, in predicting the effect of mutations which emerge in the clinic. And of course, as with the example I showed, we can understand the structural determinants of these differences in activity. So here we're looking at a different class of antibiotic, cephalosporins, and looking at 2 closely related enzymes. One can break down a cephalosporin and the other cannot. In other words, the barrier is higher to the reaction. And we can understand from analyzing the stabilization, the structures involved in the reaction, why this difference arises. And it's not a single smoking gun. It's not one interaction missing. It's a combination, a synergistic cooperative combination of interactions, including a difference in hydration at the active site. Those subtle differences are responsible for being active or inactive against a particular antibiotic. So we've gone, and again, in terms of looking at methods, so I'm switching gear away from QMMM calculations to molecular dynamics simulations. We're interested in further investigating and predicting the causes of antibiotic resistance in these beta-lactamase enzymes, these enzymes that break down antibiotics, beta-lactam antibiotics, penicillin. And we're using here a dynamical non-equilibrium molecular dynamics method, which goes back theoretically to Keal and Onsager and popularized and developed by Giovanni Cicotti. And this complements the more familiar equilibrium molecular dynamics simulation in that we take an equilibrium trajectory and we perturb it repeatedly at different points and look at the difference between the trajectories. And this allows us to assess the statistical significance of the perturbations that we see in here. So we can look at, for example, a change. It's possible to look at a change in oxidation state or to look at the effects of removing a particular small molecule bound to a protein. And we've used this to look at these antibiotic resistance questions. So what you're seeing here is a beta-lactamase enzyme. That's what the squiggly spaghetti is. And the colors represent the perturbation, effective perturbation over time. And what's interesting here is we're interested in why small molecules binding far from the catalytic site can have an effect on activity. So a so-called allosteric inhibitor binds over here, and catalysis takes place over here. And what the simulations are doing, we're removing the allosteric inhibitor and looking at the signal as it propagates through the protein. And you can see that over time, the signal is propagating from the allosteric site to the catalytic site. And we can identify the networks that connect the 2 regions. So this shows how an allosteric inhibitor can influence catalysis. And what was particularly interesting for us is that if you map the positions of variation between clinical variants of these enzymes, so enzymes which are known to have different spectrum of activities against different antibiotics, then many of those mutations lie along the pathways that the molecular dynamics simulations identify. So that suggests that the role of these mutations is to change the allosteric behavior of the enzyme, and also suggests that calculations like this can again be used predictively to understand the origins of the differences in activity between enzymes. So enzymes, they're big, relatively speaking. Coming from a small molecule background myself, they're quite big for me. And why are they big? Well, one striking correlation is that if you look at a series of enzymes, the enzymes that do the harder chemistry are on the whole larger than those that do the less difficult chemistry. So if we look at the ratio of the catalyzed to the uncatalyzed reaction, there is a pretty good correlation. Now, why is that? So a large part of that comes from the larger folding energy available to a larger structure. And as Ariel Varshall and others have shown, that allows for a pre-organization of the active site and a lowering of the barrier. Something else that is not coincidence is that this will also lead to an increase in heat capacity. You know that proteins have significant heat capacities, and that is something that nature can take advantage of, and we think is fundamentally important. As I'll show in a minute, we believe that the adaptation of the activation heat capacity is crucial in determining the temperature at which an enzyme has its optimum activity. And evolution has acted to modulate that activation heat capacity, the change in heat capacity between the reactant complex and the transition state ensemble. And by doing so, it determines the activation heat, the optimum temperature. You can see this in many natural enzymes have curved temperature dependence. So an Arrhenius plot is non-linear. And this is the old textbook picture is that this is because enzymes unfold at high temperatures. Well, of course they do, but generally for many enzymes, you see this curvature well below temperatures of unfolding. So for this particular enzyme, you can mutate one amino acid and significantly shift its temperature optimum. And that's not due to unfolding. That's explained by what we call macromolecular rate theory, which is really just iron theory, transition state theory, but incorporating a change in heat capacity during the course of the reaction. And what's also interesting, I think, is that this sort of behavior is apparent for biological systems on the large scale. So, for example, respiration from fields is not fitted well by an Arrhenius relationship, but is fitted well by macromolecular rate theory. We can do molecular dynamics simulations. These are equilibrium MD simulations where we look at the change in the fluctuations between a reactant state and a transition state. And that allows us, through simple stat-mech relations, to calculate the difference in heat capacity, so to extract the activation heat capacity. And what we find is that these simulations give activation heat capacity, which are in very good agreement with those derived from experiment. So this really does show that this is a dynamical effect, something that is modulated by evolution. And this also relates to why enzymes are big. So these two enzymes here with long complex names, they're large, and one of them is dimeric, it's two halves. And the non-reacting half of the protein plays a large part in determining the activation heat capacity. So one of the ways that nature has dealt with the question of how to aim for a particular optimum temperature is to add chunks to the protein far from the active site. So this again is why does evolution introduce mutations far from an active site? This is one of the reasons. And this is something that we can apply in the context of enzyme design and its complement of directed evolution. So the poster child of enzyme design is chem elimination, a reaction for which there are no natural optimized enzymes, but there are a number of successful designs. But the design catalysts are not very good. If you want a good catalyst, something approaching a natural enzyme, you have to carry out directed evolution, which does this mysterious addition of mutation far from the active site. Why is that? Well, one thing that was very surprising when this work was done by Adrian who's now in Bristol, who carried this work out as part of his PhD with Don Hilvert and ETH. And one thing that was very surprising is that the evolution of catalytic activity also gave rise, most unexpectedly, to the emergence of non-Arrhenius behavior to curvature in the temperature-dependent plot. So somehow, evolution is deciding that it doesn't want this to be an Arrhenius-like catalyst. Why is that? Well, again, simulations provide a route to understanding what's going on here. We can calculate the activation heat capacity, and we find that indeed we can predict the difference in the activation heat capacity between the design, which is an Arrhenius catalyst, and the evolved variant, which is non-Arrhenius. And we can look at what the changes in the structure are that give rise to the change in dynamics. And what evolution does is it introduces a correlated network, and specifically in the transition state ensemble. So this is the conformation of the protein that is responsible for catalysis. That tightens up. And I think this tightening is a signature of better transition state stabilization. And we can use the networks arising from these calculations. And what was striking for us is that the networks appear to predict the positions of mutations far from the active site introduced by evolution. So it provides a way to target those sites for evolution and design. I'll finish off very quickly by talking about ways to work together virtually. So this is a COVID target, a main protease, it's the target of Paxlovid, so it's a target for covalent inhibition. QMMM calculations have been useful in understanding the mechanism of this enzyme. We've also used interactive simulations in virtual reality to look at this enzyme. And why is this useful? Well, it's useful for two reasons. I'll skip to showing a user eye view of a simulation. So that's the heart-shaped thing here is the main protease, and within it, what Becker Walters, my PhD student, is doing here is taking the colored region, which is a peptide. This is a viral peptide. She's created a model, so it's difficult to bind long floppy molecules to proteins using screen or using routine or automated computational tools. But humans are very good at this if we can do it in 3 dimensions. And what is quite different in VR, and I will stop, is the ability to work together in the same virtual environment. So what's happening here is that Helen and Becca are in the same virtual environment, both working on the SARS-CoV-2 main protease, looking at binding inhibitors to it. But what you can't see from this video is that they were both in their own flats, their own apartments, so physically distant, different parts of Bristol. And using the cloud, it's possible to work together. We run simulations, for example, in Frankfurt, and work together in the same virtual environment. And I think that is something where there's been a lot of hype about VR for many years. But I think the potential of VR for molecular simulation is now beginning to be realized. So I will stop there, and we've actually used this in developing some inhibitors of that enzyme, which I think are quite interesting. So thank you for your time. Thank you for these guys for funding, and I'm happy to take any questions.
Thank you, Adrian, for a fascinating talk. Are there any questions? Yes, please.
In the first part, when you did this quick evaluation of the delta G, you didn't say much about the sampling. So how can you do the free energy sampling this quick way?
It's a very good question. Those are all based on umbrella sampling. So we're using an approximate reaction coordinate. The first calculations we scanned across different reaction coordinates to calculate a free energy surface. Once we had a better handle on what we felt the reaction coordinates were, then we reduced to a one-dimensional coordinate, which was a combination of different intraatomic distances. What we've done now is to move to using a string method, so the sort of method that Edina may well talk about, where we don't have to define a reaction coordinate, we have to simply define the endpoints to move between them, to do it in a less biased way, and that actually gives better results, not surprisingly, than the umbrella sampling that I
And this
talked
you
about.
do on a QMMM surface or just MM?
QMM, but it's so we're using, for those calculations, we're using SCC DFTB, so it's cheap. The barriers are in absolute terms too low, not surprisingly. But what we're able to do is discriminate between good and bad catalysts within it. And with the new project with Tom and Richard, we'll be going to higher levels, to respectable levels of density functional theory for these systems.
Any other questions?
Question from online, Sean asked general question, why does QMMM eliminate functional dependence between the two?
So it's not the QMMM that eliminates the dependence, it's the embedding of an ab initio region within the QM region. So those are probably skimmed over that somewhat in this. So we have we calculate a reaction pathway with a QMMM, a DFT QMMM method, using the sort of method we were just talking about. For example, defining a reaction coordinate, or using nudge elastic band, calculate a potential energy profile. We extract configurations from those calculations and recalculate the energies, embedding a couple cluster region within that density functional theory region. And then, so the original DFT energy profile, you have very different barriers with different functionals, but when you embed the ab initio within that, that dependence disappears.
I have a question. When you couple these QM regions, so how do they interact with one another? Is this sort of the electrostatics only, and the electron densities at your ab initio level region and the DFT level are then self-consistent, so you have to iterate back and forth?
So effectively, this would be within a frozen DFT.
Like with the frozen DFT.
But it can be extended too.
But you could self-consistently.
Yes, this is the method developed by, excuse me, Tom Miller and Frank Mamby. So it is this rigorous separation, so you can do it fully self-consistent.
Yeah. Then another question: how do you calculate heat capacity, activation heat capacities? I've never come across that.
That's a very good question. So you have to do rather extensive sampling, as you would imagine.
Yeah.
We create molecular mechanics models of the two different states. Mm-hmm. The obvious question then is, how do you represent the transition state? That can be either a physical transition state analog. So in the case of ketosteroidoisomerase, there's a transition state analog, or it can be a high energy tetrahedral intermediate, for example, which is close to the transition state ensemble. Then we look at the fluctuation within each state for multiple trajectories, so microseconds of NV simulation, and look at the difference and look at the significance of the difference between. So at first I was pessimistic about our ability to calculate these, but it turns out, I think because we're looking at differences, that again, the cancellation of errors works in our favor. But that's all molecular mechanics MD.
All right. Thank you so much. Are there any other questions for Adrian? Yes, please.
We still have time. Coming back to the last point, this is heat capacity thing. So in surface, we understand it like this. In order to react on a surface, the molecule has to bind to the surface. And then if I increase the temperature, the absorption constant gets smaller, of course, but it gets easier across the barrier. Are you thinking in similar ways to understand this?
So the heat capacity, the activation heat capacities we're looking at is the difference between the bound reactant and the bound transition state. So it's not associated with binding per se. So it's a relatively subtle difference. So this is so calculating the heat capacity for binding would, I think, be impossible in these brute force simulations. So we're looking at the difference between two complexes. So it is that, I think. So it's and then of course the question is what is the transition state ensemble? And this implies that there is a particular conformation of the enzyme which is distinct, which is responsible for catalysis. So a two-state model catalysis.
Any other questions? If not, then I would like to close this. Oh, Richard, sorry.
It's a very quick comment. Just following on from the last question, that it would be really interesting to see if this concept is applicable in other areas of catalytic science. But for example, zeolitic catalysis. I'm sorry, I may have mentioned this before.
Absolutely, and I think that several possible applications of that, but there are some, as you well know, of course, there are signatures of non-RNA today. They're often attributed to diffusion and so on in zeolites, but I think there may be non-RNase chemical behavior going on in some of those large non-biological systems as well.
Great. So then I would like to close this first session. There is tea and coffee in the Nyholm room, and we reconvene at 3:20. Thanks again to all the speakers. First session. Thank you.
Hey, George. Just let me see if you can hear me.
Okay.
Okay, hopefully, yes. Yes, so thank you very much. It's really nice to be here, and it's a pleasure to give a presentation about our work. And let me start by, I'm a bit worried that I think there's like a sound here on my computer. Maybe I need to. I'm not, I haven't set up something completely right. Anyway, hopefully it's going to be fine. So, okay. So in my group, we focus on phosphate catalytic reactions. As you all know, phosphates are some of the most important reactions. Essentially, in all basic biological systems, you would have phosphate catalytic enzymes. They would be part of the production to create RNA and DNA. In energy storage and transfer, you have these biological machines that hydrolyze ATP and then form all kinds of molecular motions. Or in signaling, we would have phosphorylation, for example, in kinases that regulate signal transaction, and when something goes wrong, and there's diseases such as cancer. So many of those are very important cancer targets. And even in metabolism, where the actual reaction doesn't have to do with phosphate catalysis, we would still have ATP that would be involved in activating these chemical processes, chemical steps in the different biosynthetic pathways. And one of the examples is the the ligation reaction that is activated by ATP. Now, when we study this using our QMMM methodology, which is very nice to present here, as is the main focus of this meeting, we have to vary not only about the phosphate groups, but very everything acid in the protein. And it turns out that some of the most important parts of these reactions are the coupled proton transfer reactions that take place together with the phosphate cleavage. And it happens to be that, during these reactions, it's really difficult to see experimentally where the protons are. So in the crystal structures or crystalline structures that we have access to, we would actually not know whether those protons are there, whether the groups are protonated and what happens with these protons. And we have even less information about these high energy transition states, what happens to the protons. But it just happens to be that what we see from our simulations, that exactly these coupled proton transfer processes are part of these rate limiting steps and are very, very important for us to really get right and try to model. Now, in addition to the proton transfer, we also have some very important other aspects that we have to take care of, and some of it is the presence of magnesium ions, as magnesium ions are almost always required for these phosphate catalytic reactions. And just like these systems are very important for biology, they are also important in disease. And here I've shown just a couple of examples where some of the drug molecules bound directly to these metal ions that are essential for the catalytic reactions. And the examples I want to talk about is, for example, HIV integrase, where some of the best drugs have been designed and they coordinate directly these metal ions, which is again classified metal ion catalytic mechanism. And these drugs are taken for 20 plus years, and the people still have major side effects against these drugs. Another example I will talk about is the prototype that we have been working on for a long time, is RNase H reaction, and as I just mentioned, there are other examples, for example, here are some of these experimental structures that were very good. Now, one example that we can do when we model these systems is we look at what happens using MD simulations, and these are, and we perform here some relatively short MD simulations to look at why the first generation HIV integrase inhibitors are really affected by drug resistance when this, for example, is very important double mutant develops in HIV. And so what we notice is that some of the it's relatively distant mutations that are here found. So like mouse doesn't work very well. So these relatively distant mutations that are here, that are not that close to the drug molecule, actually impact the stability of this metal ion binding site. And for example, they displace this water molecule that would normally be very strongly bound here in a stable hydrogen bonded system. So when we have this histidine that is one of the mutated residue, then this water molecule gets easily displaced. And even within these 100 nanosecond long simulations, you can see that the stable water molecule, which is there for the white type, and gets displaced, which is shown here by the different colors that new water molecules come in. So we see that these kinds of events affect the stability of this magnesium coordination. And the different molecules, drug molecules, then react to it differently, and the original first generation compounds are destabilized in their binding. Now, we are not able to see a full unbinding in these short-term simulations, and for that, we really need to do something more than just these simulations. And so this is what one of the main focuses in my group, to actually try to see how could we move beyond these relatively short-term simulations to something which happens actually to model, to be able to model these processes which are very important in biology. So if you would, for example, want to start our simulation, one of the best things you could do is to use, for example, Google's DeepMind protein structure, and we could use that as our original starting point for our simulations. But we would, to be able to actually model what the function of this protein is, we really need to be able to model the dynamics and the processes that actually take place. So with that, we would not be able to use DeepMind anymore, We could use our computer. And so if we use our GPUs, or maybe we use R2, even with that, we would not be able to reach those millisecond long time scales that, for example, the protein co-production changes take place. And not even with the Anchor machines from the show, we would be able to, or probably we would be able to see some of them, but we would not be able to sample it at the statistical accuracy that we would like to see. And then if we think about the catalytic reactions that take place in the order of seconds, or drug unbinding that could be in the order of minutes or hours, then we are even really far from just trying to use plain MD simulations. So what we have to do is we need to be able to sample these rare events, and we have to use some kind of enhanced sampling algorithm. This is what my group is focusing on. So here I will show you one example how we go about such system. This is our long time ago we started working on this environment analysis. And so this is a prototype to material catalytic reaction, RNA, which leaves this RNA backbone. There is this catalytic water molecule here, and the two magnesium ions coordinate the phosphate. Now, this is also an important system. It's also a part of one of the active sites in HIV reverse transcriptase. But when we first started to do these calculations, we used a Bacillus calidulans enzyme because the crystal structure was available, and that was a while ago. And so when we look at again the chemistry, we need to realize that there are associated proton transfer steps together with this phosphate cleavage. So this is the bond, the Keal bond that needs to be broken. And then the attacking water has to get deprotonated. So there is a deprotonation step. And then the leading group, which is the alcohol OH, this also needs to be protonated for it to be stable. So we have the deprotonation step and the protonation step that need to work properly. So we first looked at the different options that could happen, and we see that there is hydrogen monochloride phosphate that will make residue. Or it's also very close to this acrylic phosphate, so that will also be an option. And actually, this is what the previous simulations have found. And there are also different options for the protonation through this 2' OH, through this acrylic phosphate, or what we thought is through this conserved acetate. So when we have done, when we started these simulations and we did geonomizations to kind of sample this initially, we found that the proton transferred to this glutamate residue, so we thought that really was the mechanism that happened. And when we looked at the protonation of the leading group, then the most likely option we found was a protonation through this concept of strategies. Although we was a little bit, we were a bit uncomfortable, not I would say not uncomfortable, but we kind of highlighted that we couldn't fully explain the role of this 2' OH, because this one, it certainly changes the catalytic activity, as we have different substituents that are able to find from different hydrogen bonds. So we actually did try to use that also as a possibility for the proton transfer, but we never had a protonated water for the proton to be transferred all the way there. At this point, we modeled these two options. And to be able to model that, as we have a really high dimensional reaction coordinates of several proton transfer steps at the same time as the associated bond cleavage and formation, we use the string method in generative dimension. And so actually by both speakers. And so basically we use we implemented finite temperature string method, where we run molecular dynamics, molecular dynamics, and we kind of have a one-dimensional path along in this high-dimensional space. And this allows us to and we optimize this one-dimensional path to find single free energy path. And then in addition, we also do the Hamiltonian vertical exchange between these string images that additionally help with the sampling, and we do not add additional forces simulation. So this is maybe another example of how this method works. So we start with an initial path, which at least goes across the main biases to local minima. And as we optimize this path, we find that we eventually converge to this main free energy path that would help us to find what is the free energy barrier for this reaction. So when we, for example, apply it to this method, we find that this path can be optimized. And what we also find is that the radiant stack for this first phosphate water-oxygen bond formation is basically this coupled proton transfer is the radiant stack. So it's not just alone the phosphate cleavage, but it really is very important that this proton transfer is coupled to this phosphate cleavage. So now we decided to revisit the system, and instead of looking at this bacillus halidoranus enzyme, we wanted to look at the HIV reverse phase, and we wanted to see what happens in this enzyme. And there is one key difference here, is that instead of that glutamate residue, we now have here histidine. And this is actually the more typical situation in these enzymes. So when we now wanted to see where the proton goes, we again have our three possibilities. If we go to this oxygen of the phosphate, it could transfer to this phosphate group that we have found before. But actually, it could transfer to this histidine. And now in our simulations, what we find is that the proton transfers consistently to this histidine. If we start a path where the histidine is not in the QM region, then the proton transfers to the phosphate group above what we found before. But when we had that back the histidine into the QM region and use these optimized paths, it will still transfer back to the histidine when we continue this optimization. So we actually think that this is what really should happen in this enzyme, especially that this histidine is very, very much controlled, and its mutation to either really increase the catalytic activity or actually make it not work at all. So we see that the histidines are very highly conserved, and we were just unlucky to try an enzyme which has a glutamate instead in that specific position. But nevertheless, other residues such as this glutamate, which coordinates our magnesium ion B, are even more conserved. And actually, these residues for coordination in the lithium are even more important as their intention completely inactivate the enzyme. So when we can consider what could be, could there be really a possibility that we have protonation near the 2' OH? And so what we have done before, we tried to protonate a water molecule and see how it can reach that 2' OH and get close to the active site and close to protonating this leaving group. And so what we identified here is that there is a water tunnel which becomes really a single line water channel very close to this active site. And this allows potentially this proton to be transferred there. But when we just simply protonate a water molecule, it doesn't go all the way unless you use a large QM region. So what we learned is actually that we have used actually a too small QM region, and this was the reason that we never saw the proton transfer there. And with a large QM region, we saw, which includes some of these groups around this single line water channel, you can see that the proton can reach all the way to interact with this 2' OH. And eventually, we believe that over there it can protonate this glutamate. And when we look at our initial minimizations, what we find is that the most optimal path happens through this glutamate, which is also supported by several experimental evidence. For example, we see also that the pK of this group is significantly upshifted. It's in a hydrophobic environment. So most likely this glutamate is protonated and we have and it's also very important because this is one of the prototype reactions to a very complex glutamate. So this might be the reason they introduces here a glutamate and phosphate in many of these different pathogens. So finally, the thing that we have a mechanism which agrees with many of the experimental evidences that are not direct, but the main that this very complex protein transfer pathway really takes place this way. And the next step we would ask how this enzyme proceeds further, and how, for example, the RNA chain is now removed is yet a very complicated question and a different question. And so, for example, probably a methanion is now recruited, is more highly negative region here. And at this point, we have a really difficult task to be able to model these very complex processes. Because at this point, we don't have such a clear reaction coordinator. We would know what are the bonds that are forming or breaking. And so it is very difficult to model at this point these type of processes, and definitely not with QMMM, but even with MD simulations. So I like to now tell you a little bit of a different story, where we would like to see how can we identify such transition states, and how can we identify very important collective variables that would help us describe these complex processes. And I like to use another example, which is actually a kinase, which does use a phosphate transfer. But here we were focusing on ligand binding, and it has been a recent paradigm shift in some of the drug discovery, where it's really important to look at not just the binding affinities, but the residence time that this drug has in the binding pocket. So what is the off rate of all these different ligands? And so we decided to look at this, and we looked at, in this case, a kinase sample, which might not be the most important in terms of drug discovery, but at least we had some experimental binding off rates for several ligands, and we decided to look at them. And so these are very good drugs. So this transition state is relatively high origin. So when we used umbrella sampling simulations to model these processes, and we used a collective variable, when we looked at the initial bound pose, and we kind of identified we identified these for initial bound interactions, and using only that with a one-dimensional umbrella sampling variable, we very much underestimated this variable. And so maybe I'm going to skip why, but basically, if we don't include all the important coordinates, what we can show, even if we have a perfect sample, we will underestimate our variable. So now, to be able to identify more relevant collective variables that also capture the transition states, we would carry out these simulations where on the fly we would identify important context between the ligand and the protein, and then we would follow this unbinding process, and then we would keep on registering what are these important collective variables until the ligand completely hits binding site. And we also use this for biasing in these simulations, whenever we identify interaction here. So for example, this is one of these examples of some of the collective variables. This red distance initially is very high, then it comes to within the range where we would already use it for our biasing and for making sure that this bond is going to be broken, and then at some point then it's actually broken and it's not important anymore. So we generated different unbinding trajectories this way, several different unbinding trajectories, and at this point we wouldn't know what are the corresponding free energy barriers for these processes. These are basically very much non-equilibrium simulations. So then we use the same methods to identify these free energy paths and to find the corresponding quantitative free energy barriers that correspond to this path. But what we wanted to do is to go a step further, and we already understand what the bound pose looks like. We also understand that in our case, unbound system looks like, but we wanted to be able to characterize that transition state and to see what are those key collective variables that are really important at that transition state. And so for that, we developed a machine learning approach where we would run downward trajectories from our top of the transition state. And what we can show is that using different very simple like machine learning methods such as neural networks or decision trees, basically even on a very simple model system where we had a double line potential, that would be our make another example that would be coupled to different other potentials here with an alpha coefficient. You see that when we train the system to identify at the very, very early steps what would be the outcome of the simulations at the end. And we see that we get a 98% accuracy. And if we remove these features one by one, we can still identify the ones that are the most important ones, which have the highest correlation. And so that we can see it works very well for our data system or also the machine learning. So when we did the same thing for our analysis with our ligand, and we run our trajectory initiated from the transition state, and we just took these very, very early initial trajectories, what we see is that we actually do learn something beyond what we would know from our stream collected variable. So if we would try to decide what will be the outcome and decide on a cutoff based on our stream collected variables, we would get an accuracy of about 60%, whereas the machine learning is able to predict already at 80% accuracy, so significantly better. And so we also can ask the question, what were these important collected variables that have to decide? And we can see that they had to do with the peripherals of the protein interacting with some of the residues. So with that, we hope that maybe some of this type of analysis would be helpful in identifying what are really important collected variables in very complex processes where we can perform these simulations. So with that, I'd like to thank my group and yes, and I finished.
Thank you, Edina. And we have time for one or two quick questions. Adrian, please.
Thanks, Emma. So for your machine learning methods, how do you select the features to learn from?
So we have taken the ligand and around it like a 6 angstrom radius and included anything possible there. Ideally, we would like to also include protein content distances and far away distances as well. But at this point, we had already hundreds of them, over 100 collected variables, but we were still a bit limited. I think by now we could work with about larger numbers.
So this would be suitable for the early stages of the path.
We could, so for example, what we are now working with, what Pedro is working on, is we actually took the protein and we created kind of a grid of like several residues where this is our, let's say, coarse-grained protein, and then we looked at all possible distances within these coarse-grained components, and then used those also as collected variables. And so that could be thousands of them. Now it just turns out that our machine learning was slightly overtrained because you have just so many collected variables that are really very much similar to each other. So then our method of finding out which ones are the important ones are a bit more tricky.
Okay, thank you. If you have any more questions, then maybe you can ask Edina at the coffee break.